Maddie, if you would stand up, we'd like very much to welcome you with a round of applause. Today's forum is titled Innovative Treatment Options and Breakthroughs in Cancer Care. And I just want to say, personally, cancer has surrounded my life. I've lost most of my family through cancer. It's interesting, my husband's father died of breast cancer in 6 months. My husband's mother has breast cancer, and now my middle stepdaughter has just had her 4th cancer surgery. So it is near and dear to me that we find a cure, and I think it's near and dear to everyone in this room. A number of remarkable advancements have been made, but the cost to our society is huge. I'm not going to do that right now, but what interested me, I used to serve on something that the American Cancer Society had, which was called the National Cancer Dialogue. And President and Mrs. Bush Sr. were the chairs. I was the vice chair, and they brought together a number of major people in cancer, advocates, scientists, doctors, that kind of thing. And during that time, we heard from a doctor by the name of Brian Drucker. And during that period, the human genome and molecular biology, we were seeing many advances. And Dr. Drucker explained to us how he came to develop with a pharmaceutical company in a partnership, a drug called Gleevec, which was used for myeloid leukemia, and how it didn't attack all cells, it attacked just the bad cells. Consequently, hair didn't fall out. It seemed to be, at the time, and I don't know what's happened to it since, but very effective with that disease. And what this sort of awakened me to in my non-medical way was the fact that, it is possible to target certain things to certain cells, and much is happening in science to really change the plateau on which all of this works. And so I thought it would be a good idea to have a discussion on innovations in cancer, new things that are happening because of the human genome and because of advances in molecular biology. So we have been very lucky to actually bring together a number of very important scientists in this field. And I would like to introduce them to you and have them begin this. I will introduce the first, which is Dr. Louis Stout. He is the director of the Center for my understanding is that Senator Blumenthal just arrived. Hello, you're so modest, I didn't see you. Before I introduce the panel, I'd like to thank you for coming. I'd just like to say that I've had occasion to work closely with this new senator on the Judiciary Committee, and I found him just to be a very impressive and dedicated senator. So it's quite wonderful that he's taking the time to be here today.
Thank you, Senator Feinstein. I would like to thank you for including me and giving me the privilege of actually introducing or commenting just very briefly, because I have a committee down the hall and I'll try to come back afterward. But I want to say thank you for convening this very exciting panel on innovation in cancer. You know, we're all touched by cancer in one way or another, whether we realize it or not, whether we acknowledge it or not, and you've just described very eloquently your experience with it. Dr. Rosalind Meyer, I think, demonstrates by her story, and she's going to give you her story herself, so I'm not going to repeat it here, the importance of innovation, the innovative and pioneering groundbreaking techniques that were used to treat her in TIL at the National Cancer Institute, I think demonstrate how important these new techniques can be in literally not only saving lives in the future, but here and now. And I know that Sloan Kettering is going to be presenting. Very excited that Sloan Kettering is now going to be affiliated with the Hartford Healthcare unit and Hartford Hospital. This kind of spreading of knowledge, I think, is going to be tremendously important for the future of cancer care. And so thank you to Dr. Meyer for being here to tell us your story and your courage. You know, at the age of 56, with 3 children in their 20s, she confronted melanoma and decided she was going to fight it. And people were there to help her, including her family and the best docs in the nation. And thank you for being here today, Dr. Meyer. And thank you again,
Thank you,
Senator
Senator, for
Feinstein.
being here. It's very much appreciated. Thank you. Let me begin with Dr. Louis Stout. I hope I did that right. He is the director of the Center for Cancer Genomics at the National Cancer Institute, fondly known as NCI here, who is leading this center with the mission of developing and applying the science of genetics to better diagnose and treat cancer patients. And I might just say, since we're all sort of in the same boat of interest here, what's interesting, because of my husband's issues and my stepdaughter Heidi and the fact that her mother and her father had cancer, she didn't turn up BRCA 1 or 2, but we are absolutely convinced that it is a genetic issue, and so I'm very eager to hear what you have to say about that. You have dedicated over 25 years to research through NCI. This has led to discoveries in identifying new subtypes of cancer, which has led to improved therapies focused on personalized medicine. Your research is currently focused on lymphoma, where you're studying this cancer on the molecular level to improve diagnosis and treatment options. So we welcome you, Doctor, and please go ahead.
Thanks, Senator Feinstein. This will be a very short presentation. Some of it I gave as slides just because the genome comes alive a little in color rather than in words. But the basic idea is very simple. If you are diagnosed with cancer nowadays, by and large what happens is a pathologist will look under the microscope and they'll say the cells are in a particular geography and they're big or they're small and they're in a particular location in your body, and that's the diagnosis of cancer. We need and are transitioning from that towards a more molecular definition of cancer. And what we find is that in so doing, now we can find particular drugs that work in subsets of cancer defined by molecular diagnosis, don't have any effect in others. So this is the story I'll tell, and one that I think we'll hear from others on the panel as well. So the next slide, please. So the story I'll tell is about lymphoma, where this is these are 2 patients that would have been given the diagnosis of diffuse large B-cell lymphoma. If you ever heard of anybody suddenly, well one day and then very sick the next with lymphoma, Jackie Onassis, Kennedy Onassis, that is the kind of disease that we're talking about. A very aggressive lymphoma, which fortunately, in about 50% of patients, can be cured with chemotherapy. Good news. Bad news is we have to do more for the others. So I was quite wondering why all these patients, given the same diagnosis, some did well and some others, and maybe we could improve that by looking at the genes and their activity within cells. So if we could look at the next slide. So that's sort of next slide. On this slide, all I want to say is this is a major health burden. We unfortunately have 10,000 deaths in the United States alone from this particular type of lymphoma. Next slide. And so with the good support of the National Institute of Health at the Bethesda campus, we were able to develop what we fondly called a lymphochip. But basically what you're looking at is a single glass microscope slide. On that is 18,000 dots, and each dot is a different gene in the genome. And if it's green, what it means is that gene was very active, and if it's red, just the opposite, it's not active. So that is the way we compare tumors. They all have the same genes, but some are more active and have different functions than others. And we found that was a useful way to break this one diagnostic category into two different types. Next slide, please. And well, there's a minor type, but you can see that each column on that, it's sort of hard to see from that, would represent a different patient's tumor, and every line going across would be a different gene, and you can see that there are patterns that some tumors look quite like each other and some very different from others. And so the two common types we call the ABC variety for activated B cell, and the other is the GCB for germinal center B cell. And what we found, next slide, is that this did help us understand why some patients were doing well and not others. So the patients that had this ABC tumor, those were much harder to cure, and today, with our best available therapies, we're only curing about 40% of those patients. And the other tumor type, the GCB, is a bit more easy to cure, although we're not done with that one either. So we focused our attention on this ABC bad-acting tumor. The next slide. And so now I'm going to summarize, roughly 8 years of work on this one cartoon. And we view, molecular biologists view the inside of the cell in a sort of Rube Goldberg kind of mechanistic way, where this cog turns that wheel, and the next thing is the guy jumps into the swimming pool. It's a sort of a chain reaction of events. And in this case, the chain reaction begins from the cell surface and that protein at the top called the B cell receptor, down to a signaling pathway that promotes the abnormal survival of these cancer cells. So cancer is just the cells are proliferating too much and they're dying too little. And this is one of the reasons why these cells are kept alive. And so it seemed to us that if we interrupted this pathway, we would kill the cancer cells, and maybe in a very specific way. And then a very fortunate thing happened. I can give a little kudos to the American Association for Cancer Research. I went to talk about this work at the annual meeting before it was actually published, and after the talk, I was confronted by a group from a company, a small biotech called Pharmacyclics, said, "Have we got a drug for you? It's a drug that directly hits a enzyme within this pathway." And that was the beginning of a collaboration that led to a clinical trial that I'll tell you about some of the success. Next slide. That the drug is called Ibrutinib, and it's becoming very active, and recently is receiving approval, or will receive approval soon from the FDA for some lymphoma types. Next slide. So this, I also want to highlight, is where the initial work was done. It was at this clinical center. It's at the Bethesda campus that the Congress built for us, and it is such a wonderful resource because all the patients studied there are on clinical trials, so it's an entirely research facility. And our first patients were studied there with my clinical colleague, Wyndham Wilson. Next slide. And this is the only thing I'll say then, is this woman here was a 53-year-old that had experienced this diffuse large B-cell lymphoma. She had gone through the standard chemotherapy, and each time the tumor had come back, had relapsed. So she came to us to start taking this Ibrutinib drug. It's a single white pill you take once a day by mouth. It has no side effects. Your hair doesn't fall out. There's no other untoward effects.
No nausea.
No nausea, no GI at all, and it's like taking a blood pressure pill. And so then she came back to us 8 weeks later, and the arrows are pointing to some large lymphoma tumors in her abdomen in this scan. And then by week 8, those were gone. And then she keeps on coming back to us and keeps on getting scanned. She recently came back to us 3 years later, and there's no tumor. And all she's doing is taking this one pill a day by mouth. So she's definitely happy with her experience, and so are we. We've now finished an entire, what we call phase 2 clinical trial of this agent, where we've treated 70 patients. And overall, the response rate in this type of lymphoma, of this molecular subtype, was 40%. But the really important thing I'd like to say is that in the other types of diffuse lymphoma, the response rate was only 5%. So had we not known about this distinction of the two types of diffuse lymphoma, this would have been a somewhat unimpressive drug. We would say, okay, maybe 15, 18% of the time we're getting a response. But now we know that 40% of the time, if you have this particular type of lymphoma, you're going to respond. Many of these responses have lasted for more than a year, although I would say a note of caution, and it's the experience with all these targeted therapies, is that there can be resistance developed, and so that's a challenge. And it's as if we were saying we could cure TB with only one drug, or we can cure HIV with only one drug. We can't. So the charge to us now is to do more research to find combinations, and that's the kind of work a lot of us are doing.
Thank you very much, Doctor. Very impressive. We'll now turn to Dr. Fadlo Khoury. He is the chairman of the Department of Hematology and Medical Oncology at Emory School of Medicine. He also serves as deputy director for the Winship Cancer Institute at Emory. Dr. Khoury's work on lung, head, and neck cancers has led to new approaches in specifically targeting tumors. He's published over 250 peer-reviewed articles in medical journals and is passionate about preventing cancer as he is about treating it. Doctor, welcome.
Thank you very much, Senator Feinstein. Thank you to you and to members of the Senate Cancer Coalition. Sorry, this voice doesn't get any better, as Dr. Sawyers knows. I want to thank you for inviting all of us here and for your devotion to making progress in cancer research possible. My name is Fadlo Khoury. I immigrated to the United States in 1982 from Lebanon because I believed that we have the greatest society, the fairest society, and the society that's most likely to impact the diseases that I care about. And this is the disease that we all care about the most. That's not to say there aren't other major priorities, diabetes, cardiovascular disease, neurodegenerative disease, but certainly since the war on cancer, the National Cancer Institute has really led progress, and it's led it in an ideal public-private partnership. I myself am the beneficiary of career development support from the American Cancer Society, from the National Cancer Institute, and from the American Association for Cancer Research, which is where people like Lou Stout and I go to get new ideas and talk with our colleagues from industry freely, scientifically, to try to develop new ideas. The diseases I've devoted my career to making an impact on are lung and head and neck cancer. The vast number of these tumors are related to tobacco smoke, but we have learned a great deal from a handful of patients who seem to be increasing in number who get lung cancer, head and neck cancer, but never smoked. And that's thanks to the genomic revolution that Dr. Stout was just discussing. Today, we have the power, thanks to the Human Genome Project and innovative work by a number of scientists, to really understand and personalize cancer therapy. Now, unfortunately, for lung cancer, for all the progress we've made, we've gone from 7% of people being cured and living 5 years or longer to 16%. Still, by far the leading cause of cancer-related death. And if you add that and head and neck cancer, this costs us about 30% of all people who die from cancer. So people live longer, they live better, they have less symptoms, as you just pointed out, but they're still dying of metastatic cancer. And one of the things that our group is focused on is really trying to understand the circuitry with which cancer cells escape from targeted therapy. So never having been trained as an engineer, I've had to learn that these cancer cells are very good at going from one circuit to another. My mentor, Bernie Weinstein, who had 3 great careers, taught me to think a little bit outside the box because he coined the the phrase oncogene addiction that I've seen Dr. Sawyer's use quite a few times in his work and others. And what that means is that cancer cells become highly addicted to a survival signal from a specific gene and set of proteins. But eventually, even with the best targeted therapies, the cancer escapes. And we have really fantastic evidence for that in lung and head and neck cancer. About 9 years ago, we were working on a promising new approach to lung cancer and to a number of cancers called the mTOR inhibitors. These stand for the mammalian target of rapamycin, based on the island where they found these molecules. When we discovered that giving cancer cells certain drugs drives them to become even more addicted to that survival pathway. And we thought that was a real teaching opportunity, a therapeutic moment. We noticed that the cells could become more addicted to the pathway, and therefore, if they're more dependent, potentially more vulnerable. And that's really the focus of our work in lung and head and neck cancer. We've used small molecules, we've used viruses, which in the old days we would shoot directly into a tumor in head and neck cancers, which did show some benefit to certain patients. What we've learned is eventually the cancer outthinks it because they have so many different circuits that they can go by. It's like a complicated roadway. But thanks to genomics, thanks to the discoveries of the last decade, we have been able to start to make inroads into these diseases so that patients are living longer. I do want to end by saying that this is a unique society, and it's the only society that really has, for now, 35, 40 years, has looked at the development of the next generation and the next generation, the next generation of scholars. And I think most of us in this room who've done scientific research and trained people in our labs, in our clinics, would posit that this next generation of rising physician scientists may very well be our greatest generation, much as we tend to refer to the people who emerged from World War II to build this country in Western Europe. And I think it's crucial that we continue to nurture them, to help them move forward, because I see brighter people than me 5 days a week in clinic.
Thank you very much. I'm going to, before introducing Roz, I'm going to ask you one quick thing. You mentioned that the cancer cells become, they're strong, they become addicted. To what do they become addicted?
So the theory and some of the evidence is that they're much more dependent on certain signals that they use to survive than a normal cell. The normal cell has those proteins, but it's not as dependent on those proteins as the cancer cell. So that's why if you target something like EGFR in lung cancer,
What's EGFR?
EGFR, is a molecule on the surface of all epithelial cells, so skin cells, cells in the mouth, and cells in lung cancer. If you have a mutation there, the cancer cell is much, much more dependent on that signal, and that cancer cell is sensitive to that drug at 1/100th to 1/1000th of the dose that should cause meaningful toxicity to a normal cell. That's part of the principle I think Dr. Stout was just talking about.
Another way to explain it is that every tumor clone is a result of a long evolutionary process that starts with this genetic mutation and then adapts and changes the way it behaves as it's still a cell. It's just a fundamentally different cell than normal cells. So it has become so different and so addicted or adapted to this mutant signal that then when you interrupt it, it dies, but that same drug will have no effect on the normal cells because they haven't evolved to be so dependent upon it.
Thank you.
One of the properties, just to amplify on that, that's interesting is that some precancerous cells may have that same, it appears, may have that same over-reliance addiction, if you want, on those same proteins, same genes, same pathways.
Thank you, Dr. Curry. I'd now like to introduce Rosalind Meyer, and she has a remarkable story to share. She's a clinical psychologist, an assistant clinical professor at Yale. She's a photographer, an advocate for education and the arts, philanthropist, mother, grandmother, and the list goes on. She is also a stage 4 metastatic melanoma survivor. But she's a fighter and an ardent advocate for patients and a supporter of clinical trials. So I'd like to recognize her and say, you fight the good fight, girl, and welcome.
Well, before I even start, I just want to thank everyone. I just want to thank everyone here, particularly the doctors and researchers who, I think if you don't do research yourself, you don't really realize the degree to which experiments get frustrated, they don't work out, and the kind of determination and fortitude it takes to figure out why didn't it work this time, what can I change to make it work next time. The kind of blows to your ego of like, I think I have it, oh, no, I don't. And then, okay, well, what am I gonna do? To keep at it over long periods of time, when you believe that something is there and you have to figure out how to do it. Our doctors and scientists, we owe just a tremendous debt of gratitude for your work in all of this, because we are at an unprecedented time in this country and in the world to really cure cancer. But that wasn't the case in 2005 when I was diagnosed with metastatic melanoma.
Can everybody hear? This is a funny
Stage
room.
4, okay. When my husband Jerry looked it up on the internet, it said average life expectancy 2.5 to 4 months. I was 56 years old with 3 children in their 20s, and I was not ready to die. When I was diagnosed with cancer, I was completely shocked. I felt healthy and strong. There were no physical signs that something was the matter, except for a very small pea-sized lump below my left ear. It was an unusual presentation for melanoma. It didn't show up as a mole. By the time scans and biopsies were completed, I was told that it had already metastasized to my parotid gland, my lymph nodes, and my liver. My prognosis was grim. The story I'm going to tell you is astounding, and it represents the novel kinds of cancer treatment that lie not only in our future, but in our present. It's a story I hope will inspire you and give you hope for the future, and also a testament to why we must continue to make funding the National Institute of Health a top priority, even in the face of our difficult economy. An NIH clinical funded trial saved my life and the life of many others. When I learned I had metastatic melanoma, I was devastated. I met with a melanoma oncologist to go over all my options from no treatment to clinical trials. The conventional treatment for melanoma is surgery. Chemotherapy for this disease is mostly ineffective. At the time, there were no FDA-approved treatments that were having much success, so my doctor recommended trying to enroll in a clinical trial at NIH. I had a systemic illness. I needed a systemic cure. I had been scheduled for surgery at that point before the consultation, but after this consultation, I canceled my surgery. Since that time, 4 new treatments for metastatic melanoma have been approved by the FDA, and 2 others are in the wing, and a couple of others are in the wing. One drug called anti-PD-1 has had tremendous success with fewer side effects than other treatments in clinical trials. We need to figure out how to fast-track these successful treatments to get them to the public. It's literally a matter of life and death to many people. At NIH, my husband and I consulted with a melanoma expert who suggested that I might be eligible for a clinical trial known as adoptive immunotherapy, or TIL therapy. TIL, T-I-L, stands for tumor-infiltrating lymphocytes, and it attempts to harness the body's own immune system to recognize and kill cancer cells. At the NCI, where this experimental trial was being conducted, doctors would remove my cancerous lymph node, isolate the white blood cells or lymphocytes that recognized and were already attacking my tumor, but were not succeeding to get rid of the cancer. The researchers would clone these cells and try to grow billions of them. They would give me chemo to knock out my own immune system, and then they would return this army of cells to my body where they would fight my cancer. After a number of tests, I was found eligible for the clinical trial, and I enrolled in October of 2005. I entered the hospital and had my cancerous lymph node removed. The doctors tried unsuccessfully at that time to grow cells for the TIL treatment. While they were waiting to see if the cells would grow, they put me on high-dose interleukin-2, which is a cytokine. It's something that causes your immune system to get very hopped up and attack the cancer. It only was successful in 10 to 20% of patients in the sense that people had a response to it at all. 80% of people, or to 85%, had no positive effect. But my tumors actually did shrink. I hoped I might be one of the 4% that would get cured, but that didn't happen. Over the next 3 years, I rode a roller coaster of recurrence, multiple surgeries, but I believe that anything that gives you a chance to live another year or another month may mean a successful treatment could be developed by the time you need it. I needed such a breakthrough in August of 2008, when, to my horror, dozens of tumors appeared in my abdomen. Although the scientists had not been able to grow my cells for TIL therapy the first time in 2005, tumor tissue from my later surgeries allowed them to succeed in 2008. And I was the first person to get a particular variation of the TIL treatment at the NCI. The researchers hoped this new variation would be as successful as it had been in animal testing, but it had never been tried in humans. I read the informed consent form. It was daunting. It listed every bad outcome someone had had in earlier versions of this trial. But I knew some of the people had had their tumors disappear from this treatment, and I wanted to be one of them. One of the most moving moments for my family was the day the technicians brought in the cells they had been lovingly growing for weeks, along with a card signed by all of them, wishing me health and luck. An amazing 84.6 billion cells were infused back into my body, along with more IL-2. My children, my husband, and I did a little war dance around the IV pole. Go cells, go, I said to myself. I was in the hospital for almost a month. The treatment was very difficult, but by March 2009, all of the tumors except one had disappeared. My doctor recommended removing it surgically, so we did. The pathology report showed there were no live tumor cells in it. We were ecstatic. It's now been 5 years since I received the TIL therapy, and I continue to show no signs of cancer. I've had no other treatment than that treatment. The doctors are hopeful that the melanoma will not recur. Dr. Steven Rosenberg, who pioneered TIL therapy and is my doctor, told me recently they're now getting a 40% durable complete response rate with this treatment. When I had the treatment, it was only 15% in my cohort of patients. This was unheard of 5 years ago. Since I was diagnosed with metastatic melanoma in 2005, 2 of my children have married, both of my grandchildren were born. I was able to be there for these momentous life occasions because of our country's investment in cancer research through the NCI and NIH. Living in the times we do, there are very few of you whose lives have been completely untouched by the ravages of cancer. Some of you will have had friends or relatives who've died of cancer. Others will know people currently struggling with cancer or in remission, currently living with their cancer. Some of you will have had cancer yourselves or have cancer now. And unfortunately, the likelihood is that some of you will have cancer in the future. With each passing year, we understand more and more about the biology of cancer and the pathways that are involved in its growth. We know about tumor microenvironments and genetic mutations and biological pathways. We can analyze tumor genomes. And all of this has a direct bearing on how to mount an attack on cancer. Although we've learned so much, we still don't know for many cancers which mutations are the ones that really matter, which combination of treatments will best deliver the knockout punch to the cancer but leave the rest of our cells alone. And this is what clinical research and clinical trials are all about. You've heard about personalized medicine, genetic engineering, targeted therapies. These are all new concepts based on our growing understanding of how the body works, how cancer interacts with the body to turn off the immune attack response, and how we can turn it back on. We can defeat this illness, but only if our nation continues to fund this critical research. As a patient, I tell you now is the time. We do not have time to wait. We are making extraordinary headway in our search for treatments and cures, but too many people are still afflicted and dying from cancer every day. If you have ever confronted cancer face-to-face and had to come up with a plan to try to save your life or the life of someone you love in the face of an illness that has no successful treatment, you understand and appreciate the extraordinary resources we've created in this country at the NCI. If the cancer you have has no cure right now, no standard treatment that works, you will want the expertise and cutting-edge science embodied in this research and treatment. You will bless the men and women at NIH and at NCI and across the country who dedicate their lives to discovering new cures for debilitating, life-threatening illnesses. and you'll bless our government for having had the wisdom to make funding medical research a priority in order to improve the health and life expectancy of all Americans. Thank you very much.
Thank you very much. Thank you. Is it Renier? Oh, Renier. Next is, thank you. Next is Dr. Renier Brentjens, and he joins us from Sloan Kettering Cancer Center. He's an attending physician focused on treating leukemia. He balances research in the lab with bringing the latest concepts to trials that can directly impact patients. He's also an associate member on faculty and continues to push the envelope with how research from one type of cancer may affect another. His current work focuses on advancing treatments that target tumors using the body's own immune system. His work on leukemia is paving the way for extended studies that include other types, including ovarian cancer. Doctor, welcome.
Thank you so much, Senator Feinstein, for inviting me to this very honorable...
Is your mic on?
Yeah, it is. I think so. To this very honorable panel to be sitting on. I don't think I can beat the last talk, but I'll give it a go. 15 years ago, I was a fellow learning about medical oncology, interested in doing research, and what then was a young field of what was called translational research, where we would take stuff from the benchtop and translate it to the bedside. And with a great deal of naivete, perhaps, but certainly enough enthusiasm, I started on a project, an innovative project, wherein we would train those same TIL cells, those same T cells, the patient's own immune cells. We would train them genetically to recognize cancer cells, which they otherwise would not recognize, and to kill those cancer cells. And a very basic idea came about that we would take out a cancer patient's T cells, redirect them through the introduction of a gene that encoded for a chimeric antigen receptor, or a CAR, a receptor, that would, when expressed by the T cell, allow the T cell to recognize and kill the cancer cells when these cells were infused back into the patient. It was a very fanciful idea. It required a lot of faith, and it wasn't necessarily, it looked good on paper, but it wasn't necessarily obvious. And certainly as a very young investigator, funding from the NCI was critical to moving this technology forward, and I'd like to make a special acknowledgement for the Damon Runyon Foundation, which also had faith in this type of technology and helped fund my career at a very early stage as well to get to where I am today. So the first step was to make the receptor, which we did in the laboratory, and we could demonstrate that T cells, when they express this receptor, were able to kill tumor cells in a Petri dish. But there are many easier ways of killing tumor cells in a Petri dish than having to generate T cells for that. I very fondly remember my first great victory, and if many more such experiments happen only perhaps once in an investigator's lifetime, was when we first treated our first batch of mice that had tumor that we injected into the mice, and then we treated them with these T cells. And we wanted to see how many of these mice would live longer than the mice that were not treated with these T cells. And so I remember quite distinctly going with my wife on vacation to Ireland, and I wasn't allowed to bring a cell phone. I wasn't allowed to call the lab. That was my wife's rules. But I had 10 mice that were healthy in the treatment group and 10 mice that got the control treatment. And I knew that these mice, if they were going to get sick, were going to get sick about the time that I was in Dublin. And so I came back very eager from that vacation, went that evening to the lab, and 10 mice that we had treated with our T cells were all alive and looked perfectly healthy, and all 10 of the mice that were treated with the control had passed away while I was in Ireland. So that was the type of victory that keeps you going, and the type of astounding experiment that keeps faith and keeps you going to push this technology further, and we did. We pushed the technology further through the next 4 years to developing clinical trials. And one of those clinical trials was treating patients, adult patients, with B-cell acute lymphoblastic leukemia. This is the same type of cancer that Maddie had, who was just here, who unfortunately had to leave a little early. And in adults, certainly, as well as in children, but more so in adults, this is a disease that has a very poor prognosis, and certainly less than 30% of all patients, adult patients diagnosed with this disease, are long-term survivors. And those patients that wherein the disease relapses, the survival is less than 10% overall. And so this was certainly an area of need. These were patients that we started to target 2 to 3 years ago with our therapy. We've treated the first 2 patients on this treatment, and both patients had relapsed disease, went back into a sort of remission before we gave them the T cells, and then went on to bone marrow transplant. It was the 3rd and 4th patients that we treated who, after treatment with salvage chemotherapy, still had overt disease left in the bone marrow. 60 to 70% of the cells in the bone marrow were still tumor cells. We treated them with our modified T cells, not expecting much, but in both cases, 7 to 14 days later, we repeated the bone marrow aspirates, and there were no detectable tumors left in these patients. In fact, by deep sequencing, a highly sensitive methodology to detect for residual B-cell tumors in this situation, we were unable to detect any residual tumor cells, and these patients would go on to bone marrow transplantation. We got a lot of press attention when we published this at first, but it was obviously very gratifying to take something from such a simple concept that was still a long shot 15 years ago to something that is now being pushed forward into a phase 2 trial. Since we initially published those reports 6 months ago, we've treated an additional 9 patients. Overall, 4 of the 14 patients we've treated, 12 of them went into complete remissions and were eligible for bone marrow transplantation, which is the only potential salvage for these patients. We think that this is certainly a step in the right direction, and these CAR T cells, which Maddie herself received and had such an outstanding response to, I think are the first iteration of a type of therapy that will be expanded to not just B-cell tumors as we're treating right now, but to other types of cancer. The current iteration is, to use the car analogy sickeningly forward, is a Ford Model A, and what we're ultimately want to get to is the Ferrari, or some American brand of car. Yes. A nice SUV or something. The technology has far to go. It's a technology that's in its infancy. And for it to move forward, it will require continued funding by the NIH. And again, given this day and age, we hope that we get continued funding so that this technology can realize its full potential and not just go after B-cell malignancies, but go after a far wider array of cancers. So thank you for having me.
Thank you very much. Would you? Round of applause, please. Thank you. Thank you. Now, while you're getting ready, there'll be a microphone passed. If you raise your hand, we'll come to you, but I'm going to exercise my privilege of being up here by asking Dr. Stout a question. Lung cancer is the #1 cancer cause of death. All my life, I've always known people who have been smokers who died from lung cancer. Now, increasingly, I'm coming across people who have no history of smoking. Is this new? What is happening? Could you talk a little bit about it?
So what you're hitting on is what we call the heterogeneity of cancer, is that just as I was saying, it's almost a misnomer to talk about lung cancer. There's so many different types of lung cancer, and what happened was that this particular mutation that Dr. Corey was talking about strangely occurs much more often in folks who have never smoked.
Wow.
And
We should all smoke, ladies and gentlemen. No, don't take that seriously.
And yet they still sustain that. And we now know, if you think about the fact that our lungs are breathing all day long for our entire life, and you live near the Beltway, as I do, you could imagine that we are, even though we're not smoking, we are constantly exposing our body to the environment. And when you look at the genome of lung cancer cells, whether you've smoked or not, you have many, many more mutations in the genome than you will find in other types of cells in the body. So this is part of living here in the Beltway and maybe better in the plains of Kansas, but nonetheless, it is the part of our body that's exposed. So that is the kind of thing that causes mutations that then will, after many years, and not in everybody, will lead to cancer.
Could I amplify on that?
Yes, please, thank you.
So I agree with Dr. Stout, who put it very eloquently. But here's the very curious thing. Every day in clinic now, we see patients who, even with the newer techniques, and we're part of a 14-institution coalition that includes the NCI and Sloan Kettering, the Dana-Farber and others. So we at Emory and others, we see patients who seem to have the identical genomic drivers of lung cancer. And some do really well, and some do not as well. Now, on balance, patients with lung cancer are doing much better, but you're right, we're seeing more of these people who, as Dr. Stout mentioned, never smoked, who have these signature driver mutations. Here's the problem. It's that, as I said, they do better for a while, but we don't have permanent cures, not the way we have with chronic myelogenous leukemia with Gleevec, as you started our session very eloquently talking about. But what we do have is insight into what drives these individuals' tumors, what the patterns of resistance are, and how we can begin to target it. So what we still don't know is, other than smoking, which I wouldn't recommend to anyone in this audience, certainly not you, what drives these tumors and what allows the resistance. Once we've figured that out, I think we'll have a more systematic way, for example, to decide that someone like Dr. Meyer should get immunotherapy, someone else should get targeted therapy, someone should have a sequence. So the first step was Dr. Stout's signatures of why certain lymphoma patients do well and some do poorly. But then understanding why they do poorly, why they get the disease, that's the next step.
Thank you very much. There was a question. Anybody? I thought somebody had the mic. Oh, here we go.
Sure, I'll go ahead and start off.
Could you speak right into the mic?
Yes, it seems appropriate here to talk about the role of Congress, and I'm curious, some amazing discoveries that you're talking about, what can Congress do in their role to make your job easier, or alternatively, what can they change to make your job less difficult? Does it simply come down to funding, or are there more complex issues of regulations involved?
Well, I'll answer that. We can get rid of sequestration. I don't believe that sequestration is really necessary. Our debt is declining. We've made $2 trillion worth of cuts so far, and that's enough, in my view. I've been here for 20 years. In the early years, and Senator Specter had a lot to do with this, we had a goal to double the funding for the Cancer Institute within, what was it, 10 years? 10 years. And we did achieve that. And I think as a product of that, a lot of research has been done that would not otherwise have been done. So if we could go back to that, I guess it was the head of NIH who came before our Democratic caucus and told us the impact that sequestration was going to have on grants. And I think it was something like 50% of grants were not going to be able to be funded because of sequestration. So right then and there, you have a dimming effect on it. So that's why I say get rid of sequestration.
Can I add something? So yes, funding is absolutely critical. It was already before sequestration daunting to try to get NIH funding to do this type of research. There's a very real risk that if the funding remains as low as it is, there will be a lot of junior faculty and a generation of future scientists, enthusiastic, innovative scientists, that will never be able to get a leg up or maintain funding for their own research projects. And we may be at risk of losing those trained scientists to industry or just to the MD PhDs that just go to be fully clinical. So I think that there's a very real risk that the longer this situation maintains, the lifeline of academic research is NIH funding. And if we lose that or it becomes unattainable, there could be long-lasting effects in the research community.
Please, go ahead, Rosalind.
The kind of treatment that I had, TIL treatment, is not something that actually would have come out of anywhere except the National Cancer Institute. It's not something that's profitable. It's something that the specific treatment that I had was with my cells, So a pharmaceutical company would not be interested in pursuing this particularly. A lot of the research that happens in universities and in institutions, cancer institutions, are things that we need that pharma can then later develop, but without the scientists who can pursue this, because there's money to give them to pursue experiments and to write grants and to have them peer-reviewed. I mean, the process is very, very, very rigorous. It's about 10% of grants that get approved at this point. Do we know? Less than that now? I mean, it's hugely, it's 6, 7, 8% now. And that's just, we're missing out on so many opportunities to fund potential cures for all kinds of diseases. So, I mean, figuring out how to put money back into these budgets is really critical.
Yes, sir?
Thank you for
Yeah,
your
thank you.
wonderful comments. Last year, a ballot measure in California would have raised the price of a pack of cigarettes by $1 and generated approximately $1 billion in additional revenue, possibly another source. I know one of the criticisms was that this would be a regressive tax on smokers, but another suggestion, an alternative, is to tax the manufacturers directly.
I'm sorry, I missed that last part. Repeat it a little louder.
Certainly. The criticism of Proposition 29 would be that the tax would be imposed on the smoker, but it's a regressive tax. But an alternative for Congress and our state legislatures to consider is to tax the equipment that cigarette manufacturers use to make cigarettes, perhaps to tax the ingredients, and hopefully to prevent them from passing that on to the user. But anyway, just another possible answer for where we can identify new revenue streams to cure and beat cancer. Thank you.
Thank you. Well, I appreciate that. I don't see that happening right now. But I do know this, smoking is much less. And I think that's an enormous step forward. I know my husband's favorite film is Casablanca, and we keep watching it and rewatching it, and everybody smokes, and it's in a room with all kinds of secondhand smoke. You don't see that very much anymore. And so I think that's really a step ahead. And let me ask this question. What percent of cancer is from smoking? 90% or more?
No, I think if you look at the cancers that are very strongly linked to tobacco smoke, that's about 35%. And the other ones that have what we would call considerable evidence, it's another 25%. So the 35% would be lung cancer, head and neck cancer, esophageal cancer, and the other 25% diseases like pancreatic cancer, lymphoma, bladder. Bladder's probably level 1 evidence. So we have
From smoking?
From smoking.
How does that happen?
Well, the problem is we know how it happens with people who have bladder cancer. You know, we have the old evidence from chimney sweeps and others. The tobacco is really a fantastic drug. One could argue that the tobacco manufacturers are geniuses, not necessarily working for the best purpose, but they've developed a very powerful instrument that gets to where we'd like other drugs that help the body to get. On the other hand, there's no question that you can find the signature mutations of tobacco smoke in the lungs, in the bladder, in the head and neck. But we're getting evidence that that's true for some other diseases, including Dr. Stout's favorite disease, lymphomas. There's a higher incidence of people with lymphoma who smoke than non-smokers. We don't exactly understand.
Is that the nicotine or is it the smoke?
It's the other
Other 29 or
materials,
30
the other
products.
mysterious chemicals that are in there that directly attack the DNA and create mutations in the DNA. And those course through your entire body. They get in through the lungs, but they're excreted through the bladder. That's why the bladder also sustains this kind of genetic damage.
Thank you. Anybody else? Yes, please.
Thank you, Senator, and thank you, panelists, for being here. My name is Joe, and I wanted to ask a question regarding clinical trials, Rosalind, particularly. I've become more and more familiar with pancreatic cancer specifically, and that's been something that we've been referring many people to. And can you talk about your experiences with clinical trials and also maybe the benefits as researchers that you've come across?
Who wants to just go ahead?
I'm sorry, I couldn't hear all that. You'd like me to
very-
talk about clinical trials for pancreatic cancer?
No, just your personal experiences with like enrolling yourself in a clinical
Talk a little
trial
bit about the
and
clinical
how that
trials
helped you.
went through.
Kind of what the process was?
Yeah.
Okay, so I first went to my university. I live in New Haven in Connecticut. And so I went to Yale and found out I had stage 4 and found out that there really wasn't anything in 2005 that was having much success with stage 4. And so I looked for clinical trials. My doctor actually did know somebody at the National Cancer Institute, and so he had worked there himself, so he referred me there. I went in and had an interview, and they took some biopsies and sent me back to have some scans and tests to check my blood protein type to make sure I had no brain tumors, other things. Then I came back, and I think one of the most important things that I also just want to say is that I had been scheduled to have surgery to remove the cancers that they knew about, and that originally it was a head and neck cancer. They didn't know that I had a liver cancer, and part of the process was they discovered that I had it. And the doctors still recommended that I go ahead with the head and neck surgery and that they were going to deal with the liver later. And that made no sense to me at all. And so one of the issues is to find out before you do anything kind of what the whole story is. Because when I went down to the National Cancer Institute, they said had I had that surgery at that time, I wouldn't have any tumor that could have shown the impact of the clinical trial treatment. And so I therefore would not have been eligible for the clinical trial. And so my not having gone forward with the surgery, which was so much being pushed by many, many people at that time, was really, I see as critical to my survival. And I didn't know that until I went down and had a meeting at the NCI, and they said, no, we're going to take out one tumor and then we're going to try to grow these cells, but we need the other tumors in there to see whether this is going to work or not. And so as hard as it is sometimes to wait when you have a cancer diagnosis, you really need to get all the information before you move. It's very hard, but it's really important, and I believe that saved my life. The process itself, in terms of the treatment, are you also wanting to know about that, or just this part? Okay, thank you.
Anybody else, please?
Hi.
Hi.
This is a question for really anyone on the panel. When Dr. Meyer was speaking about specialized treatment and personalized treatment of cancer,
I don't think, I can't hear you.
hear it
You
either.
have to
Sorry.
speak directly into that mic,
Is this
slowly,
better?
and enunciate. All this marble is not great.
okay.
Good.
When Dr. Meyer was speaking about a personalized treatment for cancer, you mentioned that it wouldn't be as profitable for pharmaceutical companies, and that's why there's not many people doing research because it's not profitable. Is there a way or incentive that we could provide for people to want to do that kind of research that might be more beneficial to cancer patients?
Say that again. Is there
A way to provide like an incentive for people to do more
Go
personalized
ahead if you
research?
understood it.
I mean, the answer to that question is the NIH. So what we do at the NIH doing basic research and the earliest stages of development of therapies is to show what's possible. Then we really need the pharmaceutical industry to jump in and supersize it and make it. And so even with the T-cell therapies that we heard these beautiful stories about, now major pharma is making an investment finally, in those therapies. And so, but it was only because the academic institutions took the first step, found the leads, found the ways to go about it, and then they're more than happy to jump in and do it. So I think that we work very synergistically together with the pharmaceutical industry. We can't do it without them. They can't do it without us, and we need to keep on getting funding for the NIH in general.
Can I ask you a question here? I thought of this when Dr. Drucker spoke to us at this cancer dialogue, and he had partnered with a pharmaceutical company. What is the sort of moral or ethical compass that guides this? So the pharmaceutical company doesn't spend this money, and then the drug really isn't that effective or good. How does that work?
So I would think if you talk to the people in pharma, they are there to cure patients with cancer.
Well,
so
also
it's
make
just
money.
such a hard, and to make money, but it is such a hard problem that, and you know, we've sort of really glass half full here today. Cancer is still very difficult to cure, and we are excited by these new opportunities. But when we find that we're only getting few months of benefit out of certain drugs, we cannot settle for that, right? We have to say we've got to do better. Unfortunately, I think it is the case that it is the NIH funding and the basic research that we do, I would say, better than pharma does, that will get to these remarkable cures, and then they will take it on. So I think that I think pharma is motivated to do better for our cancer patients, but it's just such a tough nut to crack that we all have to work on it together.
Let me, if you don't mind, let me just have this discussion for a minute. 1 out of every 7 women in this room will have breast cancer, and I guess 1 out of every 7 men will have prostate cancer. It seems to me it's much easier with the prostate cancer than it is with the breast cancer. And here's a dumb question, but I've been surrounded by it in my life. Is it better to take it out now that there are all these different kinds of treatment and hormonal therapy for this? And, you know, I go to my own stepdaughter who's now had 3 surgeries for lumps that keep reappearing, treated.
Boy, we need more
So
research on this. The real revelation is that with our improved methods of detecting things that sort of look like cancer, not all of them are going to end up killing the individual, and that in fact some of our treatments that we're giving folks on things that might actually in the end be more harmful than other ways to deal with the disease. So we're seeing this in prostate cancer, some of the tests for early detection of prostate cancer. We're finding types of processes, though they're not normal, they probably won't kill the individual. And in the process of doing surgery for those things, we can, in some cases, very much harm the quality of life. And so I think this is where we need research to figure out what is the genetic signature of a bad-acting prostate cancer? What is the genetic signature of a bad-acting early breast cancer lesion? We know that some of these, that the lesion in breast cancer could be minuscule but still deadly, but then other ones that are also small will never be that much of a problem, readily cured by, as you said, hormonal therapy. So it's more research will have to get at this sort of divide and conquer idea of, oh, you have this molecular subtype, that's more favorable, will respond to hormonal therapy, and watch out for this guy. So that's where we're going. We're not done with the job yet.
Thank you. Anybody else want to say anything? Doctor?
I just want to comment on the prior question about clinical trials. I think that it's important. We're facing the same situation right now. A phase 2 trial is not something you can get the NIH to pay for. And that's currently what's going to be necessary for the FDA to allow for this to be commercialized. I think that industry is very good at reducing costs and delivering more product to more people. And I think it is commercializable, even personalized medicine is. But innovation is not a strength of industry. Innovation is a strength of academia. Innovation is a strength of going to meetings and hearing new things and trying stuff on a small scale and finding that, wow, the mice seem to be doing better remarkably. And that's also the fun part of being in academics, is you get to try out these weird ideas, and if they don't work, you don't tell anybody about them. If they do work, you get to publish them in very nice journals. And then we deliver that to industry, and then let industry go with it from there. I think that if we, in our particular field where we are working, it's cell therapies, phase 1 trials, hopefully NIH will fund it, and phase 1 trials will be done in the confines of an academic center. Phase 2 and anything beyond gets picked up by those in the industry that want to potentially make a profit on.
Very interesting. Any other comments? Rosalind, any other questions? If not, let me thank you for coming. And again, let me thank our panelists, and particularly this very strong woman on my left.
I trained at Yale. I was a resident, infant resident at Yale New Haven Hospital.